The sequence of A's, T's, C's, and G's inside modern DNA is a biological time machine. By reading those sequences, we can map the evolutionary history of life on Earth. To visualize evolutionary history, we use a phylogenetic tree. The tips are leaves, representing present-day sampled organisms. Moving backward, we hit internal nodes, where single lineages split into two, extinct common ancestors. Branches trace descent down to the root, the oldest shared ancestor. A phylogenetic tree is a mathematical hypothesis mapping time and genetic change. To build one correctly, you have to follow a strict 5-step pipeline. Step 1 is sequence acquisition. We start by downloading a set of homologous or related sequences, like the globin gene family, from a public database like NCBI GenBank. Step 2 is multiple sequence alignment. Think of this as stacking these sequence strings on top of each other to compare them, position by position. We are looking for vertically conserved colons. These are positions where the letters are matching exactly across different species, showing regions of the gene that have remained stable over millions of years. Evolution also involves insertions and deletions. To make the homologous regions line up properly, we insert dashes, or gaps, into the grid. The algorithm needs to account for these gaps mathematically. There is an absolute rule in tree building: garbage in, garbage out. If the sequences are misaligned, the mathematical distance calculations will be wrong, guaranteeing a flawed evolutionary tree. Step 3 is choosing an evolutionary model to calculate the genetic distances between your aligned sequences. This software menu shows the selection of the Chimera 2-parameter model for measuring DNA and RNA distances. To keep the math manageable, you can group these algorithms into 3 main buckets: distance-based methods like neighbor joining, character-based methods like parsimony, and model-based methods like maximum likelihood and Bayesian inference. Step 4 is tree building. The software uses your chosen algorithm to crunch the alignment matrix. It calculates the distances and translates them into a visual topology with distinct branch lengths. Let's pause here. Look at the nodes on the screen. Based on how they connect, can you identify which 2 organisms share the most recent common ancestor? It's these 2 sister taxa, connecting at the node furthest to the right. But remember, this newly generated tree is only the algorithm's best guess. Now we have to evaluate it. Step 5, tree evaluation. First, identify clades, an ancestor node and all its descendants. Next, horizontal branch lengths represent genetic distance, not chronological time. An outgroup anchors the tree's base, establishing evolutionary time. Finally, high bootstrap values indicate strong confidence in a branch split. Understanding these specific metrics separates a blind software output from a scientifically defensible hypothesis of evolution. To recap the pipeline: acquire your sequences, align the data, choose an algorithm, build the tree, and interpret the metrics. Sequences in, evolutionary history out. Next time, we'll explore distance-based algorithms. Click to keep building.